Hello, everyone. First of all, I would like to thank for the organizers to invite me to this great event. And I'm very excited to present our recent projects on COVID-19. And as everybody knows, COVID-19 pandemic is really needed some fast kind of development of the new therapy to treat this disease. So there are a number of ways to do, but practically the most promising way to do is like a drug repurposing. So basically, the conventional kind of new drug development pipeline is here, and it usually takes more than 10 years at least. Sometimes it takes 15 years to develop a new drug from scratch. But the beauty of the drug repurposing is we can definitely reduce the time. So in this diagram, it's saying 3 to 12 years, but actually nowadays there are huge efforts, particularly in COVID-19 new drug development. So this process could be reduced much further. So it can be done in a year or a couple of years. So this is one of the most practical ways. So actually many researchers and scientists are now really working on this angle. So this is the drug repurposing. And also we need to also know about this kind of virus. So SARS-CoV-2 is the name of the virus that caused the COVID-19 symptom and disease. And this virus, as you see in a number of media or papers already, is a very spiky kind of protein. And this virus actually has some subgenomic structure that's newly identified in recent months. So there are, as you know, after pandemic, there are really unprecedented kind of effort to deliver some kind of new insights about the kind of virus, but also the mechanism of this virus. So one of the outcome is here, the subgenome structure. And also there is another kind of huge effort in multi-omics space. A lot of scientists working on this domain to understand what kind of proteins actually directly interacting to this virus in our body. And one of the paper recently published in Nature identified this direct interacting proteins of SARS-CoV-2, so named as deep proteins. And this is all for the small font, but what this shows is like now we use kind of mass spec as a technology, and then we can actually experimentally measure and identify these proteins in our body that directly talking to this virus. And also some other kind of example of this kind of effort is here. So another kind of research group working on also this virus, but some other way. So they're also using mass spec as a technology, but this time they measure the expression change of proteins in our body after the virus infection. So as you can see here, at 2 hours and 6 and 10 and 24 hours after the virus infection, they measure the expression changes, and they reported also this output in Nature very recently. And as you can see, as one of the example, you can see the time on the top of the heat map. And as you can see, when you have this virus, there are certain kind of gene clusters. They are definitely highly expressed after the 24 hours of the virus infection. So this type of the kind of data set is already available, and also the information is available. So as a kind of computational drug discovery group, we are very interested in this kind of area, and also how we can contribute our expertise to kind of tackle this COVID-19. And myself and two of my very talented postdocs work together in this project, and we eventually generated SARS-CoV-2 induced protein network. So I'm going to quickly explaining what was the basic idea behind this. So firstly, as I said, once SARS-CoV-2 is infected and coming to our body, that's directly interact to some of the proteins in our body. So that's called DIP. And there is another protein group called DAP, as I showed in previous slides. So those proteins are changing the expression pattern after the virus infection. So there are two sets of the proteins, and we made kind of hypothesis, something like the deep is the kind of cause, and the depth is like consequences of the virus infection. So we know now the kind of cause and consequences, but we wanted to know the mechanism of those kind of things are happening. So what we actually tried to understand this is like, firstly, we construct the hidden layer in between deep and the depth. Of course, we're using the protein-protein interaction database, and we're pulling out all the shortest path from one of the deep protein to one of the depth protein. And then we iterate this process to build a fully connected hidden layer in between two protein groups. And secondly, we ran a kind of network algorithm on this very complex network. It's because I will show in a minute that what the real PPI SARS-CoV-2 induced network look like, but you are now seeing very beautiful, nicely drawn schematic diagram, but it's really complex. So we need some real systematic way to understand which protein is more important than the other protein in this complex network. So we used network algorithm to identify the key protein and the key pathways. And once we understand this, the next question and the last question would be what kind of the drugs actually inhibit these kind of important pathways. So we ran drug simulation on these important pathways, and then we eventually identify certain set of approved drugs for the repurposing for COVID-19. So this is the basic idea. And then this I'm going to show all the results, but if you are very interested, please go to bioRxiv because this is already available in preprint. So next thing I wanted to show is like the one, the PPI network for the SARS-CoV-2 induced network. So you are seeing this network, and as you can see, this is really big and very complex network. So it's not really informative, even though we are trying to put some colors to highlight some important pathways and proteins. So we changed slightly the way to visualize it. And we kind of get some idea from some other kind of field, but this is one of the things we can do. So we decide to put that into the circular format of diagram. And then as you can see, there are mainly three groups of proteins: the deep, the depth, and the hidden layer. And then each kind of the protein group have kind of subgroups. As I said, the SARS-CoV-2 has a subgenomic structure, so we divided this into a number of substructure in deep area. And also in the hidden area, we also checked afterwards, of course, but we identified three main kind of enriched pathways or go terms. So we calling this as a metabolism of RNA and cell cycle and immune system and some others. So we kind of categorize the proteins into each group. And another very important kind of thing is like where these kind of proteins are located in our body, our cells. So this is called subcellular localization. So as you can see on the right upper corner, this is kind of the colored kind of legend. But there are a number of things like whether this is in the nucleus or this is on the cellular membrane or extracellular regions. So we analyze those and then we put this is in the diagram. Of course, this is very busy kind of diagram, so maybe it may take some time, but I can't tell you also everything for now. But if you are interested, please go and see the paper. And then we actually draw these kind of interactions with color because this is real information. So as you can see, there are many interactions, but hopefully you can maybe see more informative ones because we use some different colors by the group. And this is just like a slide to giving them introduction about the new visualization, but actually this is real data. So after the 6 hours of the infection, we having this kind of network. But after 24 hours, we have quite different network. There are many things to say, but because of time, just wanted to say one thing. There are definite increase of the viral-related, RNA-related activities. As you can see, metabolism RNA are now talking a lot internally, but also with some other component in this diagram. And also there is a NSP8 in the deep, which is also known to be play a key role in the virus replication. So this is one of the things we identified in the key pathway analysis. And then the next step was then running the drug simulation on these key pathways. And then after this analysis, we finally identified some set of drugs for the as a candidate target for repurposing for COVID-19. And in the end, we identified 200 approved drugs by our network analysis and drug simulation. And if I say a little bit more, 40 out of 200 are actually already in COVID-19 clinical trial. And on top of this, 30 further drugs are actually already also reported in COVID-19 researches as drug repurposing candidates. So this is clearly showing the validity of our kind of prediction and hypothesis. And we further working on this. And I'm going to show in next slide. So these 200 identified drugs actually related to 148 biological pathways. And then this is really a large number, and we need kind of proper tool to understand this. So we used artificial neural network, one of the kind of machine learning AI technology, and then we assign that into each neurons, and then we train the kind of machine, and then that automatically identify this pathway into 9 important pathway clusters. Excuse me. So this is the 9 clusters, and then some of them are quite related to immune, or some of them are really related to energy metabolism. So in the end of the day, we actually successfully classify this into 2 main MOA categories. As you can see, the purples are virus replication, and the sky blue is the immune response. And then also this artificial neural network automatically assign the 200 drugs into each neurons. As you can see in the hexagon, there are numbers which indicates how many drugs among 200 are assigned to each kind of pathways. All this detailed information is also available in preprint. So this is one of the kind of mechanism of action study. And then after that, of course, the most important thing is like to validate this in silico finding and hypothesis in the experimental setting. So we are collaborating with Weber's lab in Gießen in Germany, and then we infected the cells of course with SARS-CoV-2, and then we check the replicate level in the cell when we giving this kind of short candidate drugs. So this is the results, and as you can see, we tested 5 drugs, and 2 of these, proguanil and sulfasalazine, showing this kind of efficacies. And then also we checked the kind of cell viability, and this looks fine. So we were very excited, and we decided to do the second round of the experiment with more range of those. And then we actually did, and then as you can see here, we performed on viral cells and Carlos with 2 different cell lines, and then we confirmed there is a dose response very well. So when we're giving more dose of the drug, it has more effective kind of effective way of the inhibition of the virus. And also we also checked with the biomarkers. In most recent papers, they reported very important biomarkers for COVID-19. For example, CXCL3 on the left bottom corner. So we also checked whether these kind of biomarkers are also inhibited by our drugs, and we also confirmed in this on the cellular assay. So this is going to be my last slide, and this is the kind of the summary of the mechanism of action of those drugs. And we had a further study on this mechanism, and then we can find we could find some kind of energy metabolism related pathways are highly involved. And this is going to be very important for the virus replication. And this is kind of our conclusion. So I would like to thank to my people in my lab, and also our fantastic collaborator in Milner Institute, like Kostas and Becky and Gianmarco, to helping us to validate our hypothesis on the bench, and also many collaborators who helped me on this project or some other projects. So thanks for your time and listening my talk.